We will wait for 5 more minutes to let someone else join, and then we can start today's session. Good evening, everyone. Just let me start with sharing my screen. I hope the PowerPoint is visible, right? Welcome again to this week's PMRF-NPTEL session on animal physiology. This is the week 7, and we have covered neural systems part 4. So in this particular week, we have gone through various diseases, neurodegenerative diseases of the nervous system. I have covered Alzheimer's, Parkinson's, ALS, MS, as well as spinal cord injury. We will be solving various assignment questions related to these topics today and learn a bit more about them through various schematic videos. So before we begin, let me briefly introduce myself. My name is Anubhav Chatterjee. I'm a Prime Minister Research Fellow at the Department of Biological Sciences and Bioengineering, IIT Kanpur. I will be hosting these sessions, and as you would already know, in these particular sessions we look at assignment questions from the previous year's run of this particular course and solve them in order to get better understanding of the course material as well as how to face questions in the assignment as well as the final exam of NPTEL. All of these sessions are recorded and the recordings are available on YouTube. This is the playlist that in which they are available. The link is also available in the PowerPoints that I am sharing as well as the NPTEL site. So if you have been unable to watch or attend the live session, then you can just go ahead and go through the YouTube link, and if you have any problems, then you can just address them in the comments, or the NPTEL forum is there in case you have questions later on. So first I would like to take any questions regarding this week's videos that were shared on the various neurodegenerative diseases, and then move from there. Does anyone has any questions from this week's video sessions, the NPTEL videos? Okay, so I think no one has any questions from this week's NPTEL videos. So we'll just go ahead with solving the assignment questions. So if you have attended the previous sessions, you would know how we go about this is that I will show you a question which will have four options A, B, C, D, and I will want you to put whatever you think is the correct answer in the chat, and then we can discuss about the question. So the first question for today is, which of the following is not a neurotransmitter? Islet of Langerhans, serotonin, GABA, or glutamic acid? So we have already have the answers coming in. Vishwajit says A. Jagruti Shantipi also say it is A. Achal also say it is A. Islets of Langerhans. Anyone else wants to try and answer this? Okay, so we will start with just a quick recap of what exactly is a neurotransmitter. So nerve endings transmit information at a localized spot or localized structure known as the synapse. The synapse is formed via the axon of the presynaptic neuron and a dendrite of a postsynaptic neuron. They join at what is known as synapse. In this case, there is a gap between both the neurons, which is known as the synaptic cleft. Once there is a signal that is going to be transmitted from the presynaptic to the postsynaptic neuron, the presynaptic neuron starts releasing substance known as neurotransmitters. These are essentially ligands that have that can bind to ion channels on the postsynaptic neuron. So essentially there is a signal that comes from the presynaptic neuron which causes neurotransmitter vesicles to fuse to the neuronal membrane and release the neurotransmitters into the synaptic cleft. These neurotransmitters will then bind to various ion channels present on the postsynaptic neuron and cause various activity which essentially involves to and fro movement of ions. There are essentially broadly two types of neurotransmitters, excitatory and inhibitory. If we have an excitatory neurotransmitter, then the overall result of this transmission would be an increase in the membrane potential of the postsynaptic neuron, which will make it easier for the postsynaptic neuron to a action potential and essentially transmit the signal further. But in other cases, we have neurotransmitters which are inhibitory. In this case, the postsynaptic neuron gets hyperpolarized and it is even more difficult for it to get activated or fire or have an action potential. So these are essentially the two types of neurotransmitters that are present. But if we look more deeply, there are a number of neurotransmitters. We have here a few of them. I will go through them one by one. So we have adrenaline, which is the fight or flight transmitter, which essentially helps in increasing heart rate, blood flow, and physical boost and heightened awareness in our neurons. Noradrenaline, which is a closely related molecule, which affects attention response to the same fight or flight response. Dopamine, which is the pleasure neurotransmitter, which helps in feelings of pleasure or helps in motivation and movement. People essentially release dopamine uh, whenever they feel this type of emotion. Then we have serotonin, which is the mood neurotransmitter. It contributes, it is essentially the happiness neurotransmitter, which transmits the signal of being happy and essentially helps in well-being. Then we have GABA, which is the calming neurotransmitter. It is an inhibitory neurotransmitter and helps calming of nerves in the central nervous system. Then we have acetylcholine, which is essentially a learning neurotransmitter, majorly involved in memory associated neurons. Glutamate, which is I think the most common neurotransmitter. It is also excitatory neurotransmitter, helps in memory and learning. And finally we have endorphins, which also are related to happiness and are released in the result of when you are well behaved or exercise daily. Then you have a lot of endorphins flooding your systems. What is not given here is division of whether these neurotransmitters are excitatory or inhibitory. That depends on the neurons as well as the receptors that are present, but they can be generally classified into either of those. So yeah, so these are the essentially the major neurotransmitters that we deal with. So coming back to our question, which of the following is not a neurotransmitter? So we have seen serotonin, GABA and glutamic acid all are neurotransmitters. Islets of Langerhans is the answer, in which these are essentially cells in the pancreas that release insulin and are not even related to the entire nervous system. So the correct answer is A, as you had already pointed out, islets of Langerhans. Any questions from this? Okay. Moving forward, the next question is CA1, CA2, DG or dendrite gyrus are parts of what part of the brain? So which part of the brain do CA1, CA2 and DG belong to? The options are substantia nigra, hypothalamus, hippocampus or the pre-Bodzinger complex. So you can put whatever you think is the correct option. Vishwajit says it is C, hippocampus. Anyone else? Raitha also says it is C, hippocampus. Shantipriya also agrees it is hippocampus. Anyone else? Achal also says it is C, hippocampus. Okay, let us go forward and look into it in bit more detail. So all of you are correct. CA1, CA2, CA3 as well as the dendrite gyrus all belong to the small organ present very deeply situated in the brain known as hippocampus. As you can see, it is just at the end or at the lower or what you known as the inferior portion of the brain, close to the amygdala as well as the prefrontal cortex, and is the seat of memory formation. How exactly does the entire hippocampus look like? Hippocampus is essentially divided into two parts. The left hemisphere has one part and the right hemisphere has one part, and both of them look like mirror images of each other. And if you actually look at histological section or histological uh, preparation of the hippocampus, you will see that it looks very similar to a seahorse. So that is where part of the name comes from. And if you look closely into one of the parts of the hippocampus, you will see these regions, the CA1, 2, 3 regions as well as the dendrite gyrus. Essentially, the hippocampus forms these trains of neurons. So there is one neuron connecting from the dendrite gyrus to CA1, CA3, Then CA3 neuron connects to CA2, CA2 neuron connects to CA1, and we have multiple of these essentially railways of neurons that go from one end of the hippocampus to the other. What exactly is the function of the hippocampus? Essentially, hippocampus is the seat of memory formation in the brain. So we had already gone through in the video lectures about how there was a particular case in the early 1900s of removal of the hippocampus from a particular patient who was suffering from epilepsy, and which caused no new memory to form for the person after the hippocampus was removed. This essentially was our first information that what the function of the hippocampus is. Now we have more important and well defined functions for the hippocampus. It is, as I have said, the seat of learning and memory formation. It is made out of the dentate gyrus, subculum, and the entorhinal cortex. It is where long-term memory is formed as well as retrieved. So without the hippocampus, even retrieval of long-term memory is difficult. And it also encodes a very important other type of memory, which is known as the spatial memory. Spatial memory is when you know or can put your surroundings into picture with respect to yourself. You can recognize where you are, where the doors of the room are, where the next building is. Essentially, this type of information. Or if you are going through a street, you would know where, how exactly you travel to get back to your home. So all of this information is categorized as spatial memory or the geographical memory of your surroundings. These are also encoded in the hippocampus. So coming back to our question, CA1, CA2 and DG are parts of the hippocampus. Any questions from this? Okay. Moving on. Now we will be talking a bit more in detail about the neurodegenerative diseases that we have already covered during our classes or the video lectures in the NPTEL. The first question that we will be facing today is, which part of the brain is majorly affected by Alzheimer's disease? The options are substantia nigra, hypothalamus, hippocampus, or the pre-Bodzinger complex. Which of the following is actually affected by Alzheimer's disease? You can put whatever you think is the correct answer into the chat. Kamlesh says it is C, hippocampus. Vishwajit also says it is a hippocampus. Vikyan also says it is C, hippocampus. Shantipriya also agrees that it is hippocampus. Anyone else? Anyone else wants to try and answer this? Okay, so let's go forward and look into this. So Alzheimer's disease is essentially a disease of memory formation. The major reason is that Alzheimer's actually originates in the hippocampus. So this is a schematic which shows the difference between an Alzheimer's disease brain and a healthy brain. So as you can see right off the bat, if you look at the schematic, you can see that the brain essentially loses a lot of its overall volume and as well as its weight, which is because of huge neurodegeneration and multiple accounts of neuronal cell death. So in the healthy brain, the cerebral cortex handles language and information processing. In a Alzheimer's brain, the cerebral cortex shrivels up and there is damage to areas which are used for thinking, planning or remembering. The ventricles also, these ventricles in the brain are various locations which are filled with the cerebrospinal fluid. This essentially helps the brain, which essentially delivers nutrients to the brain, and these are present in these ventricles. But in a Alzheimer's disease brain, these ventricles grow much larger than usual and are filled much more with the CSF. And finally, the hippocampus, we have a normal hippocampus on the left, whereas in the right you can see severe degradation and shrinkage of the entire hippocampus. Now if we look into like magnify and look at the neurons directly as compared to healthy neurons on the left, you can see the diseased neurons in case of Alzheimer's start dying. Essentially there is huge cell death, but it is a slow progressive cell death, and we have neurons which start dying off, so their number of connection between neurons decreases, and there are accumulation of various proteins in the neurons, as well as formation of plaques. These plaques are essentially proteins that are deposited in the neurons as well as the extracellular membrane, which are essentially the cause of Alzheimer's. These protein plaques essentially cause the neurons to die off. So as I said, the entire uh, disease progresses from very slowly from the early to the mild to the advanced stage. In the initial stage, the plaque and the protein tangles, which essentially there are deposition of various proteins in the neurons, which causes which hampers the entire neuronal function. These plaques and protein tangles form in areas of the brain used for learning, memory, thinking, and planning. This is followed by the movement of the disease to the mild or moderate stage, in which the plaques and tangles spread to different brain areas, which involve speaking, understanding speech, and sense of where the body is in relation to other objects, that is spatial memory. And finally, in the advanced stage, there is huge cell death, and the brain shrinks dramatically as compared to the normal condition. Patient may lose ability to communicate, recognize family members, and it is very difficult or debilitating, and they require huge help in handling and taking care of themselves as well. Essentially, there is complete loss of memory by this stage. So yeah, answering our previous question, which part of the brain is majorly affected by Alzheimer's disease? The answer is again hippocampus. Okay. Any questions from this? We'll be dealing a bit in detail about Alzheimer's in the next one. But any questions here? Okay. So what exactly happens during Alzheimer's? This is what the next question asks us. So what happens to neurons in Alzheimer's? The options are: the cell bodies become larger, the cell bodies start to die off first, processes become demyelinated but alive while cell bodies die off, or the processes itself start dying off. Archel says it is D, the processes of the neuron start to die off. Jagriti also says it is D, processes start to die off, which Vijit also agrees it is D. Anyone else? Okay. Shanti Priya also says it is D, processes start to die off. So we'll be looking into this in detail. So we have a video here, I will play it for you. Okay, just let me know if it is audible or not.
If we take a look at the brain tissue that's been affected by Alzheimer's,
is this audible?
Yes, sir.
Okay.
If we take a look at the brain tissue that's been affected by Alzheimer's disease under a microscope, there's almost always two common factors that we'll see, and these are called plaques and tangles. Let's just look at these plaques for a minute. Well, actually, they're called beta amyloid plaques. Amyloid is the name we use for fragments of proteins, so it's like not the whole protein, just little bits of it. And these are produced normally in the body. Beta amyloids are protein fragments that have been snipped off the larger protein by specific enzymes. So you have your neuron with this protein called amyloid precursor protein sort of sticking out through the cell's membrane. And so how beta amyloid forms is when these enzymes swing by and chop off our beta amyloid protein fragment. These little beta amyloid snippets are somewhat sticky, and they'll start to clump together, and as they clump, they mix with other molecules and even other neurons, forming what's eventually called beta amyloid plaques. Now, it's thought that these clumps of plaques, especially the smaller ones, may interfere with the cell-to-cell communication between the neurons, essentially blocking the signal at the synapse. It's also thought that they might activate the immune response as well, which triggers inflammation and also tells your immune cells to root out and destroy the disabled cells. Now, it's super important to note that this process is still not very well understood. It's especially unclear whether beta amyloid plaques themselves contribute to causing Alzheimer's disease, or if they're essentially just a result of the progression of Alzheimer's disease. So those are the plaques. Now let's talk about these tangles. Another more fancy name you might hear is neurofibrillary tangles. And these, unlike the beta amyloid plaques, actually show up inside the neurons. So usually healthy neurons are internally supported by and partly made up of these structures called microtubules. These microtubules are like tracks that guide nutrients and other molecules from the cell's body down to the cell's axon, which is usually connected to another neuron, and this is basically how the neighboring neurons communicate. So you can think of these microtubules like rails that carry a mining cart with a bunch of sweet ores and minerals, which are like the nutrients and the messages, to the other miners, which are like the other neurons. Now, a special protein, simply called tau, binds and stabilizes our rails. Kind of like how railroad spikes keep our rails in place, these tau proteins keep our microtubules in place. In Alzheimer's disease, these railroad spikes, the tau proteins, are chemically changed and basically aren't the right type of spikes. And instead of holding the tracks together, they pair with other tau proteins. And when they do that, they get all tangled up with each other. And since the tracks don't have anything holding them together anymore, what do? Well, they start to fall apart. And so our microtubules fall apart and these tau proteins become tangled. And since our tracks all messed up, this disrupts our whole transport system. And when this happens, we call them neurofibrillary tangles. Since the tangles are sort of like these thread-like fibers, or fibrils, we say fibrillary. And since they're in the brain, we say that they're neurofibrillary. And it's thought that the disruption of our rail system causes some communication issues between our neurons. And since they can't communicate or transport our materials, they eventually are decommissioned and undergo nerve cell death. Now, these plaques and tangles can be found in brains of many older people, but they're found to a much greater extent in the brains of those with Alzheimer's disease. And for Alzheimer's, they tend to start forming and spread from the cortex in a pretty predictable way as Alzheimer's progresses. The earliest areas affected are usually those associated with learning and memory, here in the temporal lobe. And as it spreads and more neurons are affected, it usually goes up to the frontal lobe, where thinking and planning become affected. And then more of the temporal lobe as speaking and communicating become affected as well. And then it kind of moves up into your parietal lobe, where your sense of where your body is in relation to objects around you becomes affected. In severe and late-stage Alzheimer's disease, these plaques and tangles have spread throughout most of the cortex. And as this happens, the cortex gets seriously damaged. And due to this widespread cell death, the brain actually shrinks dramatically. But since Alzheimer's progresses at different rates for every patient, the rate of spreading of these plaques and tangles varies a lot from patient to patient.
So essentially, just summing up what we just saw. Yeah, in the case of a healthy brain, especially in the hippocampus region, the all of these neurons are connected to one another and they fire together, and the information flows from the CA3, CA2, CA1 regions, and then we have normal synaptic connection in these situations. But in case of Alzheimer's disease, we have two types of proteins which essentially form tangles and join together to disrupt the entire process. The first are the beta amyloid plaques, which appear outside the neurons, which cause a neuroinflammation situation, and the second are the tau proteins. More recent research has actually indicated that the tau proteins act similar to prions, in which the proteins tangle amongst themselves, causing the microtubule assembly of the entire axonal bridge to collapse. This is even more dangerous once you understand that all the nutrients and proteins, as well as different cell organelles such as mitochondria, which are required for synaptic transmission of information, all come from the cell body. So as this entire railway or connection between the axons are collapsed, the entire uh, proteins or entire cargoes essentially form these huge plaques in between, and these are, as we saw, are known as neurofibrillary tangles, where proteins accumulate and cannot move any further. There is essentially a traffic jam. If once this continues to happen, the entire axon, without any access to nutrients, mitochondria or other organelles, they start dying off, and there is a degeneration which progresses from the axonal terminal towards the cell body or the soma. The first indication of this is a reduced synaptic connection between the neurons, which continues to essentially escalate towards neuronal cell death. This The primary areas where the Alzheimer's disease hits are the hippocampus, but as we have seen, this entire problem essentially starts with the hippocampus but goes to different parts of the entire brain and essentially shuts the entire brain down, causing huge mental problems as well as memory loss, and could be fatal at the severe stages. Okay, so what happens to neurons in Alzheimer's disease? The answer is the processes start to die off because of the entire rail system of microtubules being disrupted, and there is no nutrient or proteins or organelle transfer from the cell body towards the axonal or the dendritic ends, causing them to be degraded and dying off. That essentially what causes the entire cell death process to occur. And as I have said, that we now have evidence to believe that the tau proteins could actually initiate the entire process. So, any questions from this? Does anyone want to ask anything about this, or is this clear?
Sir,
Yes, go ahead.
um, signal, then what happened to Schwann cell? Those also break or not?
So, in case of Alzheimer's, the essential degradation occurs in the axonal terminal. The Schwann cells do not die off in the first initiation of the Alzheimer's, but once the axonal terminal dies off, the Schwann cells do not have anything to connect to, and they also degrade at later stages. But initially, Alzheimer's is a disease of these processes dying off. On the other hand, there are other neurodegenerative diseases such as multiple sclerosis or ALS that we will be discussing in later slides, which actually initiate from the Schwann cells or the myelin sheath covering the neurons to die off, and then we have neurodegeneration as a result. But here the neurodegeneration starts from the axonal terminals. Is that fine?
Yes, sir.
Okay. The next question is, which function is lost in Alzheimer's disease? The options are memory, motor activity, hearing, or vision. Vishwajit says it is A, memory. Achal also says it is memory. Kamlesh also says it is A, memory. Shantipriya and Begin also say it is A. Anyone else? Okay, so let us go forward. So just quickly going through the signs and symptoms of Alzheimer's disease. As I have said, the major area that Alzheimer's disease hits is the hippocampus, and the first sign and the major sign is memory loss, which is then complicated by other different things such as you misplace items as a result of memory loss. At moderate to severe stages, there are difficulties in decision making and judging because the entire the degeneration has spread to the prefrontal cortex, reduced ability in understanding and visual images when higher processing areas of the brain are affected, confusion with time and places, mood swings, repetitive speech and writing issues, difficulty in problem solving, social withdrawal, and inability to complete complex tasks. But essentially, all of it starts from memory loss, and we can trace most of the symptoms back to it. So, all of you were correct. The function which is lost in Alzheimer's disease is memory. Okay, next we move on to the next disease, which is Parkinson's. Neurons of which of the following areas is majorly affected by Parkinson's disease? The options are substantia nigra, hypothalamus, hippocampus, and the pre-Bodzinger complex. You can put what you think is correct in the chat. So Kamlesh and Vishwajit both say it is A, substantia nigra. Achal also says it is A, substantia nigra. Anyone else? Jagruti also says it is A, substantia nigra. Shantipriya also says it is substantia nigra. Okay, so let us go ahead and look at it. So again, we have a video here. I'm going to quickly play it for you. Just let me know if it is audible.
So we know that Parkinson's disease causes problems with movement.
This is audible, right?
Yes, sir.
Okay.
So we know that Parkinson's disease causes problems with movements, right? And what we see in people with Parkinson's disease is a reduction, a loss, a slowing down of movements. And this happens largely because of a loss of dopamine neurons in the brain. But you might be wondering, what is so special about these dopamine neurons? How do they actually cause these problems with movements? What's going on here? And that's actually a really great question. So let's have a look at how that all happens. So let's start off by jumping over here to the substantia nigra. And you might remember this, that the substantia nigra is the main area in the brain where we see this big loss of dopamine neurons, right? So we have these dopamine neurons neurons here in the substantia nigra. So they head over, they project to the striatum. So let's draw some of these. So here's some dopamine neurons here in the substantia nigra, and they go over here to the striatum. So they send their axons over here, and they go and they talk to neurons in the striatum. And we actually have a name for this little bundle of dopamine neurons, this little highway that goes from the substantia nigra to the striatum, and we call it the nigro-striatal pathway. Nigro because they head from the substantia nigra, and striatal because they head over to the striatum. So during Parkinson's disease, these neurons, they start to die off, right? We start to lose some of these neurons. And actually, this is really interesting. So as these neurons are starting to die, as we're starting to lose these neurons in Parkinson's disease, what we also see at the same time is this formation of these little clumps of proteins in a lot of these neurons. And these are called Lewy bodies. And these Lewy bodies start to form, and we're not going to go into the kind of the pathological diagnosis of Parkinson's disease at this point, but in what we call a post-mortem exam, so after the person has died and we're looking at their brain, these Lewy bodies are actually the main thing that we see in the brains of people with Parkinson's disease. And the really interesting part is that we actually don't know at this point what role Lewy bodies play in the disease. We don't know if they're maybe involved in the loss of these dopamine neurons, or maybe if they're actually like a protective mechanism to kind of try to prevent more loss of dopamine neurons, or maybe something that we haven't even thought about yet. We don't know at this point. And so that's kind of an interesting bit of research that's currently going on. So anyway, so back to our nigrostriatal pathway here. So more and more of these dopamine neurons die. We lose more of them, and when we've lost about 80% of them, so quite a bit, right? 80%, that's when we start to see bradykinesia and rigidity and some problems with movements, the things that are actually the physical signs of Parkinson's disease. So in order to figure out how this loss of dopamine neurons actually leads to the movement problems that we see, we have to kind of think back to the direct and the indirect pathways that the basal ganglia in our brains use to control our movements. So I like to think of the thalamus as a dog on a leash. And it needs to be on a leash because we don't want it getting all excited. Because when the thalamus gets all excited, it causes our motor cortex to get too excited. And this causes our muscles to move too much movement. Because remember, our motor cortex is what talks to our muscles to get them to move. So we keep our thalamus on a tight leash, right? So this is what we do. And we do this so it can't overexcite the motor cortex. And the basal ganglia, their job is to adjust the length of our leash. And this adjusts how active the thalamus can be. And they do this by talking to each other in the direct and the indirect pathways. So the aim of the direct pathway is to loosen our leash on the thalamus, to let our thalamus be a bit more active and chat with our motor cortex more so that we can move around a bit more. So let's say maybe you're crossing the street and the light is about to change. And you're a good person, you don't want to hold up traffic. So you need to go from walking to running in order to make sure that you get to the other side in time, right? So here is when your direct pathway would kind of kick in. So your substantia nigra would send off these dopamine signals to make the thalamus more active. And this would make you move a bit more, allow you to go from walking to running to get across the street. But when we lose these dopamine neurons in the substantia nigra can't send much dopamine to the striatum. So this means that it can't amplify the direct pathway. It can't excite our muscles even more. So we can't get that extra muscle movement to get our friend here across the street faster. And on the flip side, remember that the aim of the indirect pathway is to tighten that leash on our thalamus, and that's to reduce muscle movements. So maybe you made it to the other side and now you want to go from running to walking. So you want to slow down the muscle activity in your legs, right? So that you can slow down your steps and start to walk. So in this pathway, the substantia nigra uses its dopamine to kind of fine-tune this and make sure that we don't turn our muscle activity down too much, so that you want to walk at a good pace. You don't want to walk too slow. So the substantia nigra helps get you at that perfect pace. And it does this by loosening our leash on the thalamus, and it causes more movement. When we lose these dopamine neurons in Parkinson's disease, the substantia nigra, it can't send as much dopamine to the striatum to loosen the leash on the thalamus, right? So our leash, it gets too tight. and our muscle movements are turned down too much. So overall, with a loss of dopamine neurons in the nigrostriatal pathway, the substantia nigra, it just can't initiate more movement in the direct pathway, and it can't prevent an excessive reduction in movement in the indirect pathway. And that's why this loss of dopamine neurons in the substantia nigra causes the slowing down and loss of movements that we see in someone with Parkinson's disease. So in this video,
Okay, I hope that was interesting. So I will just go through some of the points of the video with the slide. So to explain it simply, I will just give you a bit of information. We already know that any sensory activity that is sensed by our extremities goes to the brain via the spinal cord in something known as the ascending pathway, in which the information from the sensory neurons flows to the spinal cord, to essentially the dorsal horn of the spinal cord, from where it moves to the higher parts of our brain. This information is then processed and is then sent down back via the descending pathway, and this descending pathway then connects to another motor neuron, and the motor neuron does its function. So this is essentially how exactly it functions. The substantia nigra is the part which connects the descending pathway of our brain to the motor neuron. So this is essentially in between the process, between transferring the processed information from the higher parts of the brain to the motor neurons. The problem occurs, so this is what a normal substantia nigra neurons should look like, and all of these are dopaminergic neurons, so they release dopamine as the neurotransmitter. But in case of Parkinson's disease, these particular neurons starts to degenerate. The degeneration is very similar to what occurs in Alzheimer's. There are plaques as well as tangles that form between the neurons and cause huge amount of axonal death, which essentially ultimately causes the death of the entire neuron. But this starts, as we said, that in case when the axonal terminal does not get its neurotransmitters which are produced in the soma or other organelles or other nutrients, then overall activity of these neurons starts to go down, and we have less and less release of dopamine from these neurons. And finally, this decreased dopamine essentially causes a decrease of the overall motor activity that we see, because the even if we can sense information via the ascending pathway, descending pathway is now dysregulated. We do not have a connection between our processing center in the brain towards the motor neurons that will actually do the function. So the in-between neurons of the substantia nigra starts degrading and dying off. This is essentially what causes Parkinson's disease. So which neurons or which of the following areas is majorly affected by Parkinson's disease? The answer is substantia nigra. Any questions from this? Anyone wants to ask anything from this? Okay. So I hope we have understood Parkinson's disease well. So the next question comes, which function is lost in Parkinson's disease? The options are memory, motor activity, hearing, or vision. You can put whatever you think is the correct option to the chat. Jagruti says it is B, motor activity. Kamlesh also says it is B, motor activity. Vishwajit also agrees it is B. Shantipriya also says it is B, motor activity. Anyone else? Okay. So as we had just discussed, the major hallmark of Parkinson's disease is a degradation of the overall motor skills of a person. So we'll just quickly go through the disease and the symptoms of Parkinson's. The as I said, the major part is the motor skill symptoms. First is bradykinesia, in which we lose control of our facial muscles and have a mask-like face with decreased blinking and degrading fine motor skills. There are vocal symptoms due to inability to control our vocal cords, rigidity and postural instability, tremors of hands, which we have seen multiple times in people with Parkinson's, where there are multiple tremors in their hands. This is also because of unregulated motor cortex activity, difficulty in walking and gait difficulties, and dystonia, which is also repetitive muscle movements that make body parts twist. All of this can also be combined with non-motor skill symptoms such as mental and behavioral issues, sense of smell, sweating and melanoma, gastrointestinal issues, and pain. So essentially, these non-motor uh, symptoms also go hand in hand with the motor skills, and essentially, a majority of the case are a result of motor skill degradation. So, which function is lost in Parkinson's disease? The answer is motor activity. Any questions from this? Okay. Reduced levels of blank in brain was seen to cause Parkinson's disease. Reduced levels of which particular component does which causes Parkinson's disease? The options are pyramidal neurons, dopamine, optic nerve, or glutamate. Achal says it is B, dopamine. Anyone else? Kamlesh also says it is dopamine. Shantipriya also says it is B, dopamine. Vigyan also says it is B, dopamine. So just quickly going through this again. Jagruti also agrees that it is B, dopamine. So in case of Parkinson's, we just saw that all of the neurons that are affected in the substantia nigra are dopaminergic neurons. So normal neurons, they release dopamine and they affect the motor neurons, and then we have normal movement. But in case of a Parkinson's affected neuron, there is very less dopamine being released, which causes less information transfer and movement disorders, which essentially is the hallmark of Parkinson's. But this less dopamine release essentially causes Parkinson's. Thus, the treatment for Parkinson's, which won the Nobel Prize recently, not recently, sometime back, was the treatment of Parkinson's with L-DOPA therapy. L-DOPA or levodopa is an analog for dopamine, which essentially reinstitutes this particular decrease in activity of the substantia nigra neurons. This essentially gives some semblance of motor activity back, because now there is movement of information from the substantia nigra to the motor neurons. Okay, so reduced level of dopamine in brain was seen to cause Parkinson's disease. Next we have the next disease that we had talked about, ALS or amyotrophic lateral sclerosis is also known as Lou Gehrig's disease, Parkinson's, Alzheimer's, or myasthenia gravis. Achal and Vishwajit say it is A. Lou Gehrig's disease. Shantipriya also says it is A. Lou Gehrig's disease. Anyone else? Jagruti also says it is A. Lou Gehrig's disease. Kamlesh also agrees, and Vigyan also says it is A. Lou Gehrig's disease. Okay. So yes, you are all correct. Amyotrophic lateral sclerosis is also known as Lou Gehrig's disease. This particular disorder is a progressive neuromuscular disease in which the lower motor neuron. So till now in Parkinson's we had been talking about the upper motor neurons which stay majority of the time within the spinal cord. But these upper motor neurons essentially connect the substantia nigra to the last bit of lower motor neurons, which are the neurons which innervate our muscles. So in case of Parkinson's, the problem was here, the connection between substantia nigra and upper motor neurons. In case of amyotrophic lateral sclerosis, the problem is here in the lower motor neurons, which are connect the spinal cord to the entire to our muscles and innervate them. So these are the long neurons which flow all the way from our spinal cord to the muscles in our hands or legs and have huge extensions of the axon. And here we have progressive degeneration of these particular neurons. So how exactly does this occur? So we know that the axons of the neurons are covered with a myelin sheath. This myelin sheath essentially helps in signal transduction as well as preventing any contact between different neuronal fibers, which will actually cause for them to short circuit. Without these myelinations, we have a decrease in intensity of the signal as well as decrease in the speed of travel of the signal from one end of the neuron to the other end. Majority of the times, the ALS is caused due to autoimmune reasons. The cells of the immune system starts attacking the Schwann cells that make up the entire myelin sheath and compromises them. This in turn causes decreased activity in the neurons, which in turn finally causes cell death of the neurons and causes huge motor disabilities. So ALS is also known as Lou Gehrig's disease. Any questions from this? Okay. Next question is, sphingomyelin in the myelin sheath is a carbohydrate, a protein, a vitamin, or a lipid? Achal says it is D, a lipid. Vishwajit also says it is D, a lipid. Anyone else? Kamlesh also says it is D, a lipid. Shantipi also says it is D, lipid. Jagruti also says it is a lipid. Okay. So first we'll go through the left side. So this is what the myelin sheath is made out of. So what exactly is the myelin sheath? It is a covering of the entire axon of the neuron, which is made out of different cells. So in case of the peripheral nervous system, the myelin sheath is made out of the Schwann cells. In case of the central nervous system, it is made out of oligodendrocytes. What happens is that the Schwann cell covers the different parts of the axons and form this huge bundle of sphingomyelin. The sphingomyelin layer is essentially an insulative layer which causes no transduction of ions through this particular sheath. So only exchange of ions and essentially action potential can only occur in these nodes of Ranvier, which are the gaps present between the myelin sheath. Overall, the myelin sheath has huge contribution towards increasing the speed of transduction of signal, because due to this arrangement the signal or action potential jumps from one node of ranvier to the other, essentially crossing huge distances in between where there is no requirement of action potential or ionic exchange. It also causes less amount of ions to be exchanged between the neuron and its surroundings to cause action potential, because there is only this small parts of the neuron that are undergoing action potential. And it also causes insulates the neurons, which are side by side, so that no short circuit essentially can occur between neurons, because all of these are like live wires through which electricity is flowing. You do not want both of them, which are very close to each other, to fire in between them somewhere else other than the axon. So these cells, the Schwann cells, deposit huge amount of this particular compound, which is sphingomyelin. Sphingomyelin essentially is a lipid which is made out of two components. One is ceramide, which is another lipid, what makes up a lipid. One part of it, which is a huge carbon chain attached to a nitrogen and a CH2O group, and a phosphocholine, which gets attached to this particular end, which form this complicated structure of sphingomyelin, which looks very similar to phosphatidylcholine, and essentially forms the entire membrane around the neuron, which essentially makes up the myelin sheath. So you are right, sphingomyelin in myelin sheath is a lipid. Any questions from this? Okay. So this is all I had for you today. We actually went pretty quickly through the entire lecture. So we have covered the assignment for this week. So any questions anyone has from the assignment, that we can deal with that, because we have a lot of time left. Anything that we can discuss? Okay. Then thank you for joining this week's class. I will see you next week. Thank you.